about the cocoa swollen seed virus disease. Here's my table of content, the introduction, the taxonomy of cocoa swollen seed virus disease, infection cycle, the mealybug symptoms, management practices, recommendation and conclusions, and then my references. To the introduction, the cocoa swollen seed virus disease is caused by the cocoa swollen seed virus. This disease was first discovered in Ghana in 1936, and it's a major threat to the production of cocoa in West Africa. The disease has affected places like Sierra Leone, Ghana, where it originated, Togo, Benin Republic, and Nigeria, the western side of Nigeria, where cocoa is cultivated. This disease led to various eradication campaigns in Ghana, which led to removal of cocoa trees, and this brought about a huge loss to farmers, especially Ghanaian farmers. The cocoa swollen seed virus disease is a pathogenic viral infection. The taxonomy, the order is Otevirales, family is Colimoviridae, genus, it belongs to this genus. Who has an idea of what this is? The adenovirus, and here's the infection cycle. This class of viruses are kind of very dangerous because they cause a very devastating effect to farm products. The viral protein attached to host receptor, and then this brings about the release of viral DNA into the nucleus, and then it brings about production of viral proteins as a result of RNA translation, and then this further leads to retrotransmission of genomic RNA in the cytoplasm of new double-stranded DNA genomes, and then the genomes are encapsulated with capsid proteins, releasing new virus. So this cycle makes the virus to multiply and eventually leading to the death of the plant. The vector for the cocoa swollen seed disease is the mealybug. Here is the life cycle of the mealybug. The female mealybug has 3 install stages, and then the male has 3 install stages and a prepupal stage and a pupal stage before becoming an adult. And another catastrophic thing about the mealybug is that it can produce as much as 100 to 350 eggs, and that means it can lead to it can proliferate quickly. There are 2 major species, Planococoides ungalensis. There are other species also. However, I'll be talking about this. The Planococoides ungalensis, the Planococcus citri, and then the spread occurs through radial spread. The radial spread is immobile mealybugs interlocked in trees. The mealybugs are interlocked within the cacao tree, and then the jump spread is the first install, like as you can see that the first install stage, and then it is the first install nib transported through wind. Other transmission of this disease could be via wounds, grafting, and infected seeds. The virus is also transmitted via the feeding activities of the mealybugs on infected cocoa. Here is a research work that clearly highlights the symptoms of the cocoa swollen seed virus disease. If you can see the highlighted part there, the cocoa swollen seed is genetically diverse. Different isolate strains can cause different symptoms, including transient red veins and molting in young leaves, different shades of chlorosis in mature leaves and pods, root atrophy and stunting, as well as root and stem swellings. If you look at this, here we can see the red vein banding in young leaves, and then here we see chlorosis of the leaf, and here is clearing between the major vein in mature leaf. Also, here we see the swelling of the stem, which is one of the very visible signs of the cocoa swollen seed disease. And then the dieback of cocoa caused by CSSV, the cocoa swollen seed virus, and then stem swelling of Japan infected with CSSV. Usually, after the plant has been infected, the plant dies off after 2 to 5 years, in between 2 to 5 years, if not removed. There have been several attempts to eliminate the mealybugs via biological control, but these attempts have not been very successful. First, because they have very high proliferation rate, and secondly, because there is a symbiotic relationship between the ant belonging to the Chromatogaster and the mealybug. The ant provides more tent to protect the mealybugs, and in exchange, the ant receive sugary exudate from the mealybugs. So this makes it very difficult. However, some management practices have been put in place. The major management practice in controlling this disease is the cutting down of trees, the removal of infected trees. This is a picture showing the cocoa cutting out replacement program in Ghana. The aim of this program was to remove infected trees so as to avoid further spread to areas that are free from this disease. There have also been certain regulations by the World Agroforestry Center to restrict further spread of this disease. Part of the regulation includes restriction to transportation of cocoa material between cocoa farms, restriction to removal of diseased plants without supervision by relevant authority or trained personnel, then sanitization of pruning and cutting tools with 10% bleach before use on another cocoa farm, and then restriction to cross-border exchange of cocoa materials. And also the use of rubber boots is recommended on cocoa farms so as to ensure proper cleaning of boots before visiting another farm. These things were put in place to avoid further spread by man. As we all know, that man is also a vector of this disease. Here are some other management practices. Barriers have also been created. The use of oil palms. Oil palms have been used as barrier on cocoa farms so as to create a wall that breaks further spread, since oil palm is a non-host plant. Also, the use of pest insecticides have been used. However, these have not been very effective. Biological controls have also been attempted. The Oesophila, the Chromatogaster, and the mealybugs have a symbiotic relationship. However, the Oesophila and the Chromatogaster are natural enemy. So there have been several attempts also to use the Oesophila genus to reduce members of the Chromatogaster. This has worked to an extent, but has not been very effective. So this was reported by Strickland in 1951. Also, another interesting management practice is the inoculation of cocoa beans with CSSV strain. In this research work by Amayo et al. In 2016 rather, 2 meat strains of cocoa solon soot virus were assessed for their capability to mitigate the devastating effect of the cocoa solon soot virus in fruit trials. And this research was carried out in Ghana. The meat isolate was actually successful to a very reasonable extent. Now, as you can see, the growth and yield of the cocoa progenies were improved using the meat strain, and also there were low expression of CSSV in the meat isolate. However, the immunity declined after 15 years. So another management practice put in place is the somatic embryogenesis. The somatic embryogenesis is a tissue culture approach to develop disease-free clonal propagules from infected cacao trees. And I found this to be very interesting because it involves the use of somatic cells to create disease-free propagules from the cocoa trees. Also, as you can see, the use of clean planting materials and resistant varieties have also been recommended. There have been various breeding programs and varieties like the Cocoa Research Institute of Nigeria. There have been new varieties of the Crentisi 1 to Crentisi 8 of new varieties, and then there have been crossbreed between the F3 and the F2 and F3 Amazon, and these have to an extent showed a bit of resistance to this disease. Our recommendation and conclusions: destruction of infected trees surrounding plantations within 25 meters rather than the recommended 10 meters. Now, according to the Cocoa Research Institute of Ghana, it is recommended that infected trees should be destroyed, and then trees within 10 meters should also be destroyed. But I really think with the proliferation rate of mealybugs, the distance should be increased, probably like 25 meters. That might probably work better. Also, feed with high infestation rate should be burnt and used to cultivate a non-host crop. If the infestation rate is high, rather than removing trees and planting another one, I would recommend that these trees they should be burnt and then used to cultivate a non-host crop, because there have been situations, according to literature, there have been cases where trees were removed and then new trees were planted, but after a few years, the new trees became infected. And that was what led to the use of barrier trees and also probably increasing the distance. But I think that rather than use the same field that have been infested to cultivate cacao again, it would be better and more economical to use them to cultivate non-host crop. Now, I would also recommend that alternate host of mealybugs within and around cacao plantations should be destroyed. Other plants that host these devastating insects should be destroyed within and around the cocoa plants. This will prevent further proliferation. Also, destruction of Crematogaster moth tent. Like we saw earlier, that the Crematogaster, and there's a symbiotic relationship between the Crematogaster and the mealybug. So rather than destroy the mealybugs and fight against the mealybugs and ignore the Crematogaster, preventive measures should be taken against the Crematogaster family, or the genus Crematogaster also. Because according to Strickland, Strickland's research, it was actually observed that the population of Crematogaster had a kind of positive correlation with the number of mealybugs present on cocoa farms. And then I would recommend also that biological control research should also be invested in. There should be more biological control research. Plants and insects that release chemicals that repel mealybugs should be worked on. There should be more research working and looking into plants that release phytohormones and chemicals that help to repel mealybugs. This will help take away the point of using chemicals to fight against mealybugs, and then it will also help to conserve the environment. And here are my references.
Thank you.
Thank you for your attention.
Any questions? Go ahead.
Yeah. You mentioned that there is recommendation for the destruction of cocoa plants.
Yes.
Is it that, did you think or you read it as 25 meters?
And that's part of my, if you look at this, these are part of my own recommendations.
Okay.
Now, the recommended distance used by Cocoa Research Institute of Ghana is 10 meters.
Okay.
So I feel like instead of use 10 meters, why not increase it? Since we know that they have high proliferation rate. So I think it will work better if we increase the distance.
Won't you be putting into place the spacing of the cocoa when it is being planted? Because for them to recommend the 10 meters, the spacing of the cocoa planting will be put in place.
Well, I think the prevention of the disease is much more important than the number of plants we have on the farm. If not, we are going to continue to allow further spread of the disease, and that will be more devastating.
All right, I have a question. You said field with high infestation rate should be burnt and cultivated with alternate plants that do not host the Crematogaster or mealybugs. Yeah. have you considered the environmental pollution that might cause? If you recommend burning of the whole field of for plantation farming, is that sustainable practice?
Well, if you look at the rate at which this group of virus proliferate, I think this is part of my recommendations anyway, not according to literature. Now, if you look at the way they proliferate, oftentimes it is usually recommended that you burn infected plants, because when you cut down an infected tree, there is still probability that if the infected tree is not properly taken care of, there can still be further spread.
Yeah, it is true that pathogens can lay in the soil for quite a number of time, but burning an entire field, yeah, it's different from burning an infected tree. Yeah.
Do you want to develop a bit on that? What did you say that pathogens stay under the soil?
Yeah, may lie there fallow for some time.
Okay, we're talking about the virus, right?
Yeah.
That's not really what.
Okay, no, not virus. Talking about fungi and
All right.
bacteria.
Well, let's focus on
this
this
one.
particular
Okay, all right.
subject.
Okay, it's all the same. For a viral disease, if you burn the field, how would that really?
Ask him.
No, it's his personal recommendation. I think it's a welcome one anyway.
All right. Well, we're making questions, right? Not comments. Okay. And okay. Was that the end of your question, or was something else you wanted to ask?
No, that's it.
Okay. Any other questions?
I've got a few. Can I ask a few?
Go on.
So on the first slide, you showed a map where the disease is located.
Okay.
Did you miss Ivory Coast, or the disease is not present in Ivory Coast?
Okay.
Do you know?
Ivory Coast.
Yeah.
Well, I think I most likely must have missed Ivory Coast, but there were no very rigid mention of Ivory Coast in many literatures as much as these other places.
Okay. And
I was there last week. There is, I confirm.
so other questions is like, you talked about the virus, the Badna viruses. Are they DNA, RNA virus? Do you know?
The what?
The Badna virus?
Yes.
Because is it the DNA, RNA virus?
The Badna virus. Is it a DNA or a RNA virus?
Yeah.
Well, I think I should be able to have a clue from this. I want to believe that it's a DNA virus.
Okay, and if it's a DNA virus, there's a possibility of it being integrated into the host genome.
Being integrated
Yeah.
into the host, yes.
Like we discussed in the lecture.
Yes.
Does this happen for CCSV?
Well,
CSSV?
I want to believe that it was the approach that was used for, it was part of the approach that was used for the, hold on, for the severe, for the mid-strain isolate. I believe it was part of the approach that was used for the mid-strain isolate because
So-
the virus was inoculated into cocoa. As the mid-strain of the virus was inoculated so as to create a kind of immunity.
Yeah, but inoculated is different of being integrated into the host genome,
Okay.
like we discussed before.
Okay.
Yeah. So do you know how this cross protection works?
I have to read much more on that.
Okay. No, just a quick question. So on your recommendation about the 25 meters, increasing to 25 meters
Yes.
to this point, what would be the cost involved to the farmer by doing that?
Well, the cost will mean reduced yield because you're going to have less cocoa plants on the field. But I think it is more better than having to destroy your cocoa plant within 2 and 5 years if infected.
I have a question as well. You said that one of the strategies which is interesting is to use barrier plants.
Yes.
Yeah, that will, well, so that will make a non-host wall.
Yes.
But what do we know about the host range of this virus?
About the host range.
Does it only affect cocoa trees or does it affect any other crops?
No, it affects some other crops.
Yeah.
Yes, I think I have a list here. This virus affects banana, it affects grape, and citrus, and probably there could be some more.
This very virus or
Yeah,
this virus or is it bannaviruses in general?
like virus in general.
Right. Okay.
Yes.
But when we're talking about this particular virus, CSSV,
Okay.
does it affect any other crops or any other
The
plants?
cocoa solan virus disease.
Yeah.
Well, I think it is specific to cocoa. That's why it's actually so tagged.
All right.
Yes.
I don't think so.
Okay.
It's a little bit of a gap in research. We
Okay.
don't.
Okay.
It has not been fully studied.
Fully studied. Okay.
And before making barrier crops, you need to actually know if they're going to be host, right?
Okay.
And that's not very well understood at the moment. One last question: the mealybugs, can they fly?
What?
The mealybugs, can they fly?
Can they fly?
Fly. They have wings.
No, I didn't read about mealybugs flying. I sincerely did not read about mealybugs flying in
Yeah.
literature, so
Because you were talking about only the mealybugs walking, right?
the mealybugs.
How do they spread the virus?
Okay. There's radial spread and then the jump spread. The radial spread is a mobile mealybugs interlocked within the trees, and then the jump spread is
Yeah, I can read, but what do you understand of that? If there's one tree that is infected in a field,
Yes.
and there's a healthy tree next to it, concretely, how does it get infected?
Now it can be infected by wind, and also when trees get interlocked, because
Yeah,
it could probably spread the infection too.
right.
Yes.
But how does it happen exactly?
How does it happen exactly?
Yeah.
Well, I want to believe that it could be through probably movement of the mealybugs from
How
a
do
tree
they
to
move?
another.
How do they move?
How do they move?
Yeah, they fly, they crawl, they run.
Okay. I think probably start with a crawl.